So what I wrote in my column for this issue is about informative censoring, which I've learned about relatively recently as a potential source of bias in a number of the oncology trials that we read every day. I thought it was, and still think it's a really important topic for all of us to improve our ability to read oncology trials critically and understand their strengths and weaknesses. And I think this particular topic is a weakness of a number of clinical trials that we read and might be a potential source of bias. And so it's something for us all to be aware of and be on the lookout for. To give credit where it's due, I'll kind of cite a couple of papers that have been my source of information on this. Most of them are co-authored by Dr. Tannock, and you see at the bottom of the slide here, two of the papers that I read that were good sources of information for me and for those who want to learn more about this topic. And there's many others, but I would say those two papers there would be really good starting points. To illustrate what this is, and this is a figure from the JAMA Oncology paper listed. These authors created a fake trial where patients were randomly assigned between a placebo and an experimental drug. And shown here is the plot, the Kaplan-Meier plot of the progression-free survival curve, showing really no difference between the two treatments. The hazard ratio 0.97, the confidence interval crosses 1, and so this is a non-significant difference, no difference in PFS. This problem of informative censoring bias occurs when progression-free survival is the primary endpoint, and it really arises when there is a differential between the toxicity-induced drop-off rate or discontinuation rate of the two treatments. That's the key features to look for is the endpoint progression-free survival and was there a difference between the arms in rate of toxicity-induced dropout. So if you see those two features of trial, that can lead to this informative censoring bias. So move over to the panel on the right. All they changed, fake model here in these curves, they changed the drop-off rate where the experimental drug increases toxicities. And so patients dropped off of the trial at a higher rate than those of the placebo arm. And what that actually leads to here is an apparent improvement in the progression-free survival of patients getting the experimental drug, even though there was no improvement in clinical benefit. There's no clinical benefit to the patient. This was just a difference of drop-off rate that led to this apparent improvement in progression-free survival. Well, the reason is that the patients who drop off of the trial before they get to a response assessment get censored, so they're not included in this analysis. And so you've then excluded the patients who have done poorly. The analysis then becomes not by intent to treat. That is, you're not analyzing all the patients that enrolled on the trial at the start. You're analyzing only the patients who have made it to their first response assessment. And that becomes a problem because then you're not dealing with analysis by intent to treat, and that is introducing bias into your analysis. So that's what informative censoring is, and it can lead to apparent improvements in benefits of a drug when in fact there are no improvements in benefits. All you've done is create toxicities with the drug. And as I've kind of learned about this and applied this knowledge to a lot of clinical trials that are out there in oncology, I've found a lot of examples of this, and it makes it challenging to interpret results of the trial. And I will say these are not just obscure trials found in obscure journals. These are trials published in the most prestigious journals, leading to FDA approvals leading to drugs that are on NCCN guidelines. And yet one could debate, if you really take this informative censoring into account, whether patients are deriving clinical benefit from some of these drugs and treatments. So when I'm reading a paper with PFS as the primary endpoint, and many of our randomized oncology trials do have PFS as a primary endpoint, I first am looking at the discontinuation rate of the two arms of the trial. And I'm looking for, is there an improvement in the PFS in the experimental arm? And is there a higher rate of discontinuation due to toxicities in the experimental arm as compared with the control arm? Many of these trials, especially those being reported out fairly early, will say that overall survival results are immature and we don't have quality of life analyses yet. And so this scenario right here, where you really need to look out for this informative censoring and consider whether the improvement you're seeing in PFS was more due to this higher rate of discontinuation of the experimental arm due to toxicities. Now, another way that these authors propose in some of their papers is to look at time to treatment failure as a form of sensitivity analysis, defined as it's similar to PFS, so you're looking for disease progression or death as events, but then you also add as events any discontinuation for treatment toxicity or discontinuation due to patient preference. This is kind of, in my mind, the same as saying, well, we're just going to assume the worst, again, as a sensitivity analysis, and assume that everyone who discontinued treatment for toxicity will call that event. And so you're assuming basically that every patient who drops off has a progression event. So, the problem with these is that this endpoint is not considered to be a surrogate for overall survival, but it does eliminate this problem of informative censoring. The FDA does not prefer this as an endpoint, primary endpoint for studies, so it generally should be used as a sensitivity analysis. The FDA has favored the use of PFS as the primary endpoint for oncology studies. But this can help you understand whether there's a problem with informative censoring in the analysis here. So something for us all to think about as we critically read the literature.